Welcome to the second day of the ECCB 2020. I have very few announcements to make. So remember, today is our last day, so we will be finishing around 5. Second announcement is remember to use the different communication tools that we have enabled for the conference, including Rocket Chat, the Meeting Hub, and the different communication tools for each session. Third, remember we will have during the long break the opportunity to interact with the different exhibitors and sponsors of the conference, including the career fair. With that, I would like to let Mark to present our keynote speaker today. Thank you.
Thank you, Salva. Welcome everybody, benvinguts to the second day of ECCB. It is a great pleasure for me to introduce our speaker today. He comes to us from Germany, Fabian Theis. I just happen to have known Fabian for our interactions into the Lifetime initiative that Genevieve yesterday introduced to all of you. And for those who didn't realize, yesterday we published a couple of very important documents for this initiative. One of them is a natural perspective, where Fabian contributed significantly to the work of Lifetime, as many others did. So, but before we let Fabian talk, that's why we are here to hear, let me tell you a bit about his background. Fabian is a mathematician and a physicist, so he has a 2 degree. He's also what in Germany called a doctor, which means that he actually got 2 PhDs, one of them in physics, and the second one in computational science in Granada. That was a surprising thing. I didn't know Fabian was in Granada doing his second PhD.
Beautiful place.
Certainly. Then he did a lot of traveling around the world. He was again in Spain, Japan, US, Germany. And about 15 years ago, I believe, he started his own group at the Max Planck Institute in Göttingen. That where he was for a short period of time, and then he moved to Munich, where he is now, at the Helmholtz Center where he's a director of the Institute for Computational Biology. At the same time, he holds the chair of Mathematical Modeling and Biological Systems at the Department of Mathematics at the Technical University in Munich. He has won many awards, including the ERC starting grants from years ago. So we kind of doubt his research has been evaluated many times and very positively. But of all the things, what I want to highlight of his career is that he's a guy who doesn't hide in the lab. What I mean by that is that he has contributed to many things outside his lab. So his work and his passion for science goes beyond what he does in the lab. So for example, he's been founding coordinator, director of many things, including the Single Cell Omics Germany initiative, the Munich School of Data Science, and the Helmholtz Artificial Intelligence Cooperation Unit. Again, he's a great contributor of the Lifetime, and we are very happy to have him here today talking about modeling cellular states and dynamics in single cell genomics. Fabian, the floor is yours.
Yeah, thanks, Mark, for these very kind words. And of course, also to you and the organizers for this fantastic opportunity to speak at one of, I guess, the biggest meetings in our field. So this is a great honor. And I'm a bit sad that we cannot physically meet up. I guess we've heard this a lot of times in beautiful Barcelona. But on the other hand, I just want to say this again, congratulate to the organizers as well, that this, I think, timeline type of system that you've been setting up for sort of getting us electronically through those days was working out quite a bit better than I've seen at other conferences. So I really enjoyed that, given that we can't actually meet up. I hope we can do this in a year or something like that. So yeah, thanks for having me. I want to talk briefly about, well, maybe somewhat briefly, about what we do in single cell genomics. So I call this thing modeling cellular state as well as dynamics. And I stress the modeling part. I guess many of you still remember that our field has gone through, I guess, a different set of renamings and of course, sort of touching fields at some point, you know, sort of this very sequence-based things initially being a big thing, structural bioinformatics coming up, then systems biology being a very big topic with modeling, then the whole machine learning aspects. I tried to point out a few avenues where I think the single cell field is playing into that, particularly with its big data sets. And my name is Fabian Theis, and I'm interested in single cell genomics and dynamics. So let me give you a brief overview. Single cell biology, of course, is one of, I guess, is the founding thing for cell biology. I mean, you know, the microscope was the thing that actually has been used to find cells. So it has been around for quite some time. This was actually how I went into the whole single cell biology field, by looking at time-lapse microscopy. So that case usually can look at many cells, but only very few factors at a time. This is from a fantastically named review from Jan-Philipp Juncker and Alex van Oudenaarde. They called Every Cell is Special some time ago. And of course, this is in contrast to the omics biology, at least at the time, where we were observing always high dimension, potentially unbiased cellular state via transcriptomics and other epigenomic variants of that, but we took ensemble averages. And only over the year, with development of a series of techniques such as high dimensional RNA-seq, then of course amplification and then isolation towards single cells, we came into a regime where those two things sort of merged, and we can observe now many genes in many single cells at the same time. Potentially not yet over time, but these things coming up, and Mark was mentioning lifetime as one of those avenues where these things are starting. And there's of course now all kinds of new fun things starting, such as spatial transcriptomics, this tracing, multi-omics, that are a big topic at the moment. Today, I want to speak about that field. I just want to highlight, for us being now here from the computational side, that I think this is really an exciting opportunity for computation biology and machine learning in general, and I think has been really contributing to the field over those past, I would say, 5 years quite intensely. This is from a recent CCI, Jan Zuckerberg grant initiative, where a whole bunch of methods have been funded just for analyzing single cell genomics. And I think if you go to singlecelltools.org, I guess you have more than 200 or 300 different aspects. Even I think just for pseudotime inference, already 70 or so different methods have been proposed. So it's fun because you have a lot of cells and you can do something with it. Why is that working? Well, we all know bulk genomics often has been seen as the smoothie of looking at essentially averages, right? Averaging over more than 100,000 cells, getting transcript out in a very robust fashion, sort of ignoring then variations from states such as cell cycle, but of course also different cell types. And the idea of single cell genomics is sort of pick away this fruit salad and see what's going on. And I guess one of the seminal techniques after microfluidics was really this focus on the fact that you can actually capture with this droplet RNA sequencing single cells in those water droplets that are embedded in oil, and then do your library prep and so on. And so essentially the cellular tagging in there, and then you aggregate afterwards, you do a normal RNA-seq run, but then you have a single cell tag for each of these things. And essentially then you can quantify those transcriptomes in large scale fashion. This has been around for some time, not the droplet-based one, but it has been already 2013 noted as Method of the Year for Nature Methods, and quite recently, just this winter, now the multi-omics, so essentially the genomics and also parallel measurement one has been put forward. It's one of the next big steps. And I should really I really want to highlight that this is becoming a big data set. In early times, efficient paper with just a few cells being published more than 10 years ago. And now we're in the regimes where very routinely we get tens or 10 or more thousand cells, essentially transcriptomes, very sparse ones, but still 10,000 transcriptomes up to 1 million. And of course, the question is, what do we do with change? In this regime, we have still this, what we used to have in bulk genomics, this small and large p problem, which, for example, for learning regulatory interactions always needed very simple linear methods, regularization, and so on. In this regime here, this is something where you can really start now trying to leverage large-scale machine learning to potentially understand the correlation and dependence structures. So I think it's really exciting and a great time to ask those old questions in computation biology, such as what drives a cellular state? How can a cell make decisions? How does it decide whether it wants to differentiate, proliferate, or die anew? how do we do these things? So let me just briefly give you on one slide an overview of the basic workflow before I explain or demonstrate one or two methods that we've been doing in the lab. So, you start with a sequencer, usually get some data, you pre-process them, and then, I sort of somewhat nonchalantly put this here in one word. Obviously, there's a lot of things involved in those pipelines, but essentially we end up in count matrices where nowadays, as I said, those could be quite wide, not as tall as they used to be. Potentially many of those count matrices now, because we can do this across batch effects, across conditions, and we can ask about those. We usually do some batch correction, therefore. In early days, just a single experiment was fine enough to find groups of cell types, but nowadays you want to potentially compare conditions. Typically, a low-dimensional visualization of this 20,000 dimensional transcript space is done initially with linear methods, just looking at PCA of this high-dimensional space, but more recently, of course, also trying to include those nonlinearities, and early t-SNE plots, UMAP nowadays being super popular at sort of unrolling this Swiss roll that maybe makes up this transcriptional space. Then you do some clustering very commonly. You find groups in this high dimensional space that you very often then associate with cell types or cell states. And I'm sure many of you are aware how complex this discussion of what is a cell type has become, and is being brought up again and again at many meetings. You then need to annotate those things, and in contrast to other fields, because you redo those data sets again and again, there's no simple standard yet for really generating those annotations. So people are still struggling to maybe potentially really strongly carry those over and do it in a robust fashion. There's no super clear ontology yet, because of course people do find, again, new substates and so on. And you might maybe look for more complex structures in the data, lineages, branching topologies, and so on. And then you do also differential expression. My lab has contributed to some of those aspects, and maybe before I say how we contributed, let me just highlight again, because this is now single cell resolution, those clustering aspects were very different than you would have in bulk. Also simple things such as differential expression, you'd think you can just use a bulk method. Well, maybe not, because if you had ensemble averages and you're just comparing, let's say, condition 1 versus condition 2, then sure, you can test with t-test or something a bit more fancy, negative binomial or something like that. You could test whether there's a shift across conditions, but maybe there's also a change in variance, or maybe some more complex pattern. And for this, there's also need for developing methods. So I think for all of those aspects, you can develop methods, and people have been doing so. What we've been at some point in the lab realizing was that the people in the lab do very different things. At the time we switched already to Python, we didn't have some easy framework for putting things together. So actually just simply for the lab has become a very useful thing to set up a framework, how we develop those methods and can put them together, working on a certain sort of shared common input format, which we called AND-data at the time. And we put this in this tool, single cell analysis in Python, ScanPy, which we're very lucky to become one of the most popular tools in the field. We downloaded more than 250,000 times, and people really sort of using that. And it's been, I think, fun to see this community grow, and a lot of people contributing and working on that. And there's been some extensions towards epigenomics recently as well, together with the Kulomitachi lab. So this is the analysis part. What do you want to do with these things? Well, let me take a step back and go back to my introduction where I spoke about systems biology, where we have different ways of approaching problems. And I guess one of the very common one is we look at networks. We're kind of trying to understand this complex data by seeing how patterns interact, how genes regulate each other, things like that. Sort of on the more statistical side, then we have machine learning approaches, and sort of on the very mechanistic side, on the sort of the other spectrum of this scale versus interpretability axis, if you want, you have dynamical models. And I guess sort of classical systems biology has been focusing on those dynamic models, machine learning, maybe being very sort of statistical, but maybe not so easy to interpret. And I think that's a very nice a sweet spot to be found for each of those different methods. And I think now that we have this high-dimensional readouts generated by single-cell genomics, and this possibility of also then perturbing, also in large-scale fashion, I think we can ask these questions. And you which of those models fit to what type of analysis. And what I want to do today, I want to show one of those very small scale methods that we've been doing. It's a really dynamic model, but doing this genome-wide, thereby not being small scale anymore, but not strongly coupled. And then on the other hand, one of those machine learning approaches to learn those latent spaces. Before doing so, let me show one of the key example applications though. And this is something I think many of you would already have seen, but just stick with me just to lay the foundation. Now that we have those high dimension gene expression spaces, one of the goals could be we want to identify a cell that is differentiating in a developmental data set, for example, or in adult human blood that's being regenerated all the time very quickly. What you do is you embed those cells in a high dimension transcript space, right? Here's one example that we've been doing early on together with the Götkins lab from the University of Cambridge, where we've been looking, in this case, it was still not full genome-wide, it was things like qPCR across a few thousand cells in blood development. If you just do a bi-clustering, you see some set of genes popping up, but you don't see that much structure. The reason being that the high dimension transcript space maybe needs to be ordered in a different fashion. And typically doesn't look like those sort of randomly uniform patterns, but potentially, you know, those cells, they can't go into all states. There's some tight regulation that happens in biology. So they somehow live on what we kind of picture as a submanifold of expression. This, as maybe developmental biology often calls Waddington's landscape, you know, this hilly landscape where a cell has some potential that rolls down, makes decision. This could be, for example, those progenitor cells that then go into two downstream lineages. And the question is, could we arrange cells along those trajectories? This has been a problem that a lot of people have been looked on early on. I guess particular tools such as Monocle from Cole Trapnell, as well as Mandalus, beautifully named from Dana Pear, being one of the first, where you essentially take often some things like random walks on this landscape and try to connect those. We contributed a tool called diffusion pseudotime that sort of systematically approaches those random walks, also with branching properties on this manifold, and essentially allowed us to order cells according to an average behavior of a random walk going through these things. And if we do this to this particular data set, what you actually see is that now you get a very nice arrangement where you can actually see much clearer patterns. So you still get this cluster here as you saw before, but you see now a sequence of things that are along this lineage being turned on or turned off. And then in this case, this was, as I said, for early blood development, you can actually interpret many of these things. You see a branching point and so on. So this ordering of cells according to lineages has been very popular. There has been a lot of comparisons, as I said, more than 70 tools being made, and then big benchmarks for these data sets also being put forward. And one of our tools, Paga, was actually doing a nice job on that. So what has the lab been doing in those past few years? We have been essentially extending those pseudotime methods towards really deep lineage estimations across organisms. We've been actually trying to incorporate also real time to see then how populations diffuse across real time points in those distributions. We've been really focusing on how to do data integration and then test for those batches. We actually have a big benchmark out that compares many of those batch correction methods just from bioRxiv. And we've been then applying this to different things, particularly here at the center, people being interested in metabolic syndrome, diabetes particularly. So we've been generating atlases of intestinal and pancreatic endocrine cells and applied this to also in drug perturbations. And more recently in this lung atlas, or in this human cell atlas efforts, focusing on contributing to a lung atlas, both in a mouse as well as in human, and then also looking into perturbations for that with really fantastic collaboration partners. We've been basically able to apply our tools to great data sets. And in the crisis this year, we've been then also able to leverage this lung atlas and look at, for example, COVID-19 receptor genes being expressed in cell type specific fashion, or then look across population variation to see what's going on there. So I think it's really interesting to use those tools to apply this to large data sets and then look for association patterns. In this case, first variation within a single subject, but then here, of course, also variation of single cell expression across the population. There's sort of added layers. And I think Jean-Pierre already yesterday was nicely hinting at this multi-scale behavior. At some point, we of course need to be able to couple those layers of variation. So this is a bit of an overview of what we've been doing. As I said today, I want to sort of make this bridge of a small scale dynamic system, and then go to a large scale. This small scale one is again this pseudo-temporal ordering that I mentioned before. It's a nice thing, but it just looks in similarities. So it's like if we look at this snapshot from a physical process called gravity, right? We can kind of see what's going on, but in physics, we have something more than just position, right? In physics, in order to describe a system, if we really want to understand the mechanistic behavior, we don't only have state, we of course also have the impulse. So we'd like to have those arrows. And well, people have been coming up with this concept. It's called RNA velocity. This was, I would say, one of the seminal papers of the past few years from Julie Lamanno, from Linderson and Karchenko labs, where they had this fantastic idea of looking at ratio of spliced to unspliced reads. It has been a bit around in bulk, but they've been doing this in single cell genomics, and they realized that across measurement techniques, there was a certain ratio of reads that were actually also being unspliced or not yet processed. So they came up with this very simple dynamical model, and that's why I was speaking about the modeling aspect, which I think for the systems biology guys was really fun, of just essentially having transcription, then measuring those unspliced reads, doing this all just per gene, no coupling, then splicing degradation. So you have those three rates. You can write a very simple equation for that, just classical 101 biomathematics. And you can look at, for example, the splice kinetics across time, things is being turned on, that's being turned off. And if you then look at those steady state ratios, you can actually estimate those rates in this dynamical system. So essentially, if you plot spliced versus unspliced, you get this football type of behavior of first a process being upregulated and then being downregulated again. So in this regime, sort of there's more unspliced than spliced reads, right? So the thing that the gene is just being transcribed, being made. So the arrow essentially of this, so the differential of this change of gene expression is pointing up and there's pointing down. And if you just look at the steady states, you can estimate those robustly. What we realized though is that with this just steady state mode, you ignore a few things. For example, if a process is being regulated earlier than just going in steady state, happening all the time, you might ignore that. And then also the transcription dynamics might not be fully identifiable, so the process might be a tad too simple to put up. So what we've been doing, we've been extending this. We call this single cell velocity via dynamic modeling, SCVelo. We work by Volker Bergen, a PhD student in the lab, where we've been essentially writing up this dynamic model, but not going in steady state, but just, this is a simple model. You can still solve it in closed form, but trying to estimate those reaction rates, as well as those cell-specific latent variables, which we don't know. We don't know those time points, right? So this is like a clustering model. It's like an EM type of thing. You have latent variables. You want to estimate the joint likelihood. And the model can be done like this. A cell could be either in an on and off state or in a steady state. And we can write up then an overall likelihood that has then those two constraints that we observe, those U and those S, according to this kinetics with those latent rates here. And we then just estimate it via EM. It's not just because, of course, we have to do this for 20,000 genes or because we filter maybe for 1,000 genes at the same time. But the approach is very similar. So first, you allocate those switching points. You tell them basically if you're you estimate those Ks. Once you have estimated the K, you assign the time points, and then you update the model parameters and then just iterate it. So just standard M. There's a few tricks that Volker made to make this really efficient. And when we do this, then we can really estimate those hypothetical steady state that's not directly the steady state, because those processes usually stop early, and we can do this now in a sort of more fine-grained fashion with this dynamic model, and we can estimate those arrows. We can also see that this thing then behaves and is sort of more robust, also the dynamical version versus those older tools. And there's been a few additional features that I won't speak about because this has just been published, but you can also, now that you have a likelihood-based model, you can do all those fun things that we know from stats, where we can really now, for example, find those most dynamic genes. We can rank them by likelihood. We can look at model fit. We can see which genes do actually fit to the model, which ones not, where we might even want to modify the model because maybe some state-specific things are happening. We can actually also learn a shared time across those different TIs that are first gene-specific, and we can also test stochastic models and so on. But let me maybe show you what you can actually do in practice. In this case, we look at a data set from dentate gyrus, neurogenesis, essentially after birth. In mouse, what you see is here those progenitor cells that form both this granulocyte as well as this astrocyte lineages, a few steady state cells. And if you now learn those overall dynamics, what you get is you get first a set of arrows per gene, but then you have those arrows essentially in a 20,000 dimensional space, and you project them down with your typical UMAP and then average those arrows, and you see those dynamical flows. What is nice, if you look at our dynamic model, it actually behaves more robustly than the steady state model, this classical one that I contrasted with. For example, in this Cacharetius, where there shouldn't be any differentiation happening, this thing actually doesn't show arrows. Dynamic model behaves nice in this GABAergic neurons, where they should be actually flowing to this later time point here. In those cells, dynamic model stays more constant, doesn't point out. And overall, you can also check for consistency scores that this is doing a good job. Now, what's interesting, you can then look at a set of dynamic factors, and then you see that those transitions you do actually make a good job, and model fit looks nice, except for some few outliers that I will talk about in a minute. So now you can interpret essentially those parameters in a more robust fashion. Quite recently, we've been actually focusing a bit to understand what happens with these outliers that I showed you before, and we've been trying to now actually extend this very simple model that fits a joint rate across the whole transcriptional space by going into multiple kinetic regimes. This is out here on the GitHub page, differential kinetics, where we've been then, once you have a clustering, can allow fitting different type of rates for each of those different spaces. And we can actually find genes or transcripts where you have actually multiple kinetic regimes. Then you can try to actually see if you can explain for those by the different setups. So I think it's an interesting way to pick up interesting genes. Let me show another example where we've been looking at pancreatic endocrinogenesis. This is a collaboration with the Heiko Lickert lab and Annika Böttcher in particular, where we've been looking into a venous fusion mouse line, sorting them for those NVF positive and negative cells, mix them, and then actually do single cell RNA sequence and cross multiple time points. I just show the latest time point. What you see here is essentially development of the endocrine lineage in the pancreas, where those NGN low sort of progenitor cells, they do some type of sighting, and then they transition towards the alpha, in particular the beta lineage at the time point being the strongest one expressed, and then also epsilon and delta cells. And if you compare this to the steady state model, for example, you don't see here the nice cell cycle behavior, which you would expect, and you actually see enrichment of S to G2 factors here, which you don't see in the steady state model. You see this complex branching, which is not nicely reflected here, and also typical velocities do play a better role. So that was very nice for us. So this is published, and I encourage you to look at that. What I want to tell you briefly about now is that we actually extended this model. This is a work by Marius, also together with Volker and others, trying to interpret those vector fields. Let's say we have now, and you can use SCVelo, there's other tools, I guess, being developed. Let's say we have a vector field. Maybe you can also at some point now measure it with labeling techniques. What do you do with this high dimensional vector field? Well, currently, we typically project it to 2 dimensions and look at those arrows. There should be more to it, right? And we should be able to do something with those high dimensional arrows. So our question is, can we interpret and analyze those single cell vector fields in a more robust fashion, and maybe give really quantitative measures for that? We try to do this with what we call cell rank. This is a collaboration, I should mention, with Dana Pear's lab from Memorial Sloan Kettering in New York. And really fantastic interaction and super exciting, essentially building upon her work, Palantir, for the non-vector case. Essentially what we're trying to do is, for each branch in a branching process, we want to assign a probability based on these vector fields where the cells flow. We want to find stable populations, both root as well as final ones, potentially also intermediate ones, and we want to understand cell trends. How do we want to do this? Well, the idea is very simple. Let's say we have our graph that describes our low-dimensional manifold that sort of lives in this high-dimensional expression space. We in addition have those velocity vectors. So what we can come up with a transition tensor, then propagate a random walk forward, as you would do with this PageRank technique that you might know from Google. And if we now propagate this all the way back, we get the root states. If we propagate it all the way to the final, sort of all the way towards an absorption state, we get final states, what those cells can be. And we can now, because we have this average across all those random walks, give probabilities which direction they go, just by analyzing essentially those transition matrices. It's a bit more fancy thing than just doing eigenvalue decomposition, because this works in high dimensions, but it goes along those ways. So it's, as I said, it's similar to PageRank. What we in reality use, it's called generalized Perron cluster analysis. And if you apply this to this pancreatic endocrinogenesis data set that I showed you before, excuse me, this case has been flipped around, but you see again the major process here towards the beta cells, a little bit the alpha cells. You can now generate a cell fate map, and you get here your roots as well as the target cells. And you can get here the probability for this case, for example, going towards beta fate, towards alpha fate. So you can basically at each point in the space now saying which direction you want to go. And you can then, for example, also see those more subtle states that in this low dimension projection you might not see, that actually tells you there's an enriching direction of epsilon, for example. And in addition, you can now look at, because you have now this alignment along pseudotime, you can now take a pseudotime and plot those probabilities of different factors for each of those lineages. So you see those trends, for example, which factors are being driven upwards in a beta cell fate, and you can project this on this latent space. In addition, if you want to sort of summarize the latent space, you can use this graph approximation technique, PAGA, that we've been doing before, and you can give your sort of uncertainty maps at each of those states, whether you go towards epsilon, beta, and so on fate. So I think that's a very high dimensional summary statistic that helps you understand those latent spaces robustly. All right, so this was for the dynamic modeling part. So this was, we can use this for lineages, we can use this for transitions. I think it will be fun to explore this to really understand regulatory motives, but let's come from the other direction. Let's come from the really statistical side. Can we maybe find more robust tools to understand this latent space that are not yet mechanistic or anything like that, but that are sort of very descriptive? And one of the classical techniques I think that all of us learn in, I don't know, stats 101, I guess, is called principal component analysis. What it does, it sucks up a gene by cell matrix, high dimensional matrix, squeezes it down, blows it up again, makes the output as similar as possible to the input. Now, if all of this was linear, this would just be PCA. This case, it's called an autoencoder. It's a tad more complicated. So of course, right, high dimensional techniques, we really want to push now neural network based approaches for that. So we use multiple of those processing layers, add nonlinearities here, and then we get an encoder that squeezes things down to the spot, like a decoder that blows it up. But essentially, it's something like a nonlinear PCA. If you haven't seen this before, I think that's the way to think about things. What we've been doing, and this is a work by Lucas, now professor in use, and Gretchen from now working with Reeves lab at Broad, where we've been essentially extending this squared loss cost function towards a negative binomial, also in this case zero inflated one. And this case, we've been basically able to look at this cleared output that goes through the bottleneck and essentially gives you a denoised version. And we can show that if you apply a proper noise model, this works much better than just make mean squared error. What's nice, because it's a neural network-based technique, this scales like hell with the number of samples, as you would expect, performs well in contrast to other methods. And for me, though, it's not this application of denoising that was the most interesting part eventually, but what I really liked is that in those latent representation, the bottleneck layer, we do see those plots that I showed you before, those two-dimensional U maps or T-SNEs more or less. But in this case, it's not as if there's some sort of parametric, a non-parametric method that gives you slow dimension, but it's really a function that generates this thing and on the fly makes it look as if it explores the full variation in the latent space. So we've been asking, is this bottleneck layer, does this maybe learn a latent representation that you can do more things with? And that's of course a popular trend in representation learning, a big topic in machine learning. We've been trying to explore this ever since. One application that we've been doing, we call this ACGen last year, where we've been trying to now add a perturbation on that and try to see how this perturbation unfolds in latent space. In this case, for example, here you see a data set two-dimensional visualization of a stimulation versus a control, in this case just PBMCs. And what we did is we left out now one subpopulation of stimulated ones and try to see if we can recover it by latent space arithmetics. Okay, we just basically added this shift vector in latent space, and indeed we're able to recover that very nicely. And we've been trying to carry this a bit forward. At the time, you know, this ACGen was just focusing on essentially one perturbation at a time, but now we try to overcome this limitation of this autoencoder. In this case, we use also rational autoencoder to actually allow for multiple conditions. So the model is as follows, just a standard rational autoencoder, just with now a conditional part added to this, such as SCVI has been doing for some time. So you have this encoder, you then sample from the, so you then sample from a mean and a variance representation. You have your latent one, you add the condition again, and you blow up it in a decoder again. The likelihood, as you know, I think you saw already the L. Actually, I shouldn't call it likelihood, because it's just a lower bound to the likelihood. It's one of the key contributions, right, of those VAE estimation can be written up like that, and now we can extend that constraint by essentially adding a layer where we want to make sure that the densities actually are very similar within one condition and could be then different. We add this called maximum discrepancy layer. This is a poster that Mohamed is actually presenting at this year's ECCB as well. I encourage you to look at that. But if we do that, we now add essentially this MMD loss to the cost function, and we can take now any condition and transform it. So in contrast to this latent space arithmetics that we've been doing before, we can actually do this type of transitions, and then we can estimate effects robustly. So this is a nice tool for us that we have been now ever since trying to explore in a bit more broader fashion. So you've seen this idea of atlas generation, right? And we can now have, we have these reference atlases. What can we actually now do with those tools? Well, you can add perturbations on top of it, but ideally you'd like to do it as in genomics, where you don't want to sort of for each experiment again and again reassemble your data. You don't want to again recluster. You don't want to do this all over again. So ideally what we want to do, once we really have set up this human cell atlas generating cell type maps for each of the different organs, where we want to map now our own data on top of that without losing variability, but also without needing to recluster the original data set again and again. We want to make this simple, and we want to make essentially those learned maps accessible without people having to have this high performance GPU-based computing infrastructure and so on. This is a problem that people have been picking up for some time. I guess particularly John Marioni and Raul Satia's lab have been one of the first to really point this out strongly, essentially trying to have a reference here, now add a query data set on top of that and try to see how this performs. But those methods at the time, though, most of it was reanalyze all the data at the same time. What we try to do here in our most recent thing that we just put on bioRxiv a few weeks ago, we call it single cell architectural surgery, or SCArches. We try to now do an integration to a reference. You want to query a data set to a reference without needing to reanalyze reference data. For this, because we're now working in this regime, in this mode, in this set of tools of deep learning, we thought, can we use transfer learning efficiently for that? So for this, let's take a set of public reference. This could be a single one, could be multiple studies. Just as you saw before, we train a rational autoencoder, essentially learn a latent representation of that. We add those reference labels here to make sure that we know where the cells come from. We can sort of take into account variation, remove batch effects and so on with that. And then we get a model repository. So we don't want users to redo this again and again. So we store those weights as well as the architecture in a model. This could be just hard-coded, but in this case, those weights we can upload to a Zenodo archive that gives you DOIs and so on. And now is the goal, we want to transfer this model across studies, potentially even organ species or particular states. Let's try to do this. So let's say we have a query data set. We don't want to reanalyze the old one, but we want now to add those query labels to this reference data set. And the issue is, of course, the query labels haven't been seen by the reference. So this could be a new batch effect, whatever. So we need to extend our architecture. So we need to add additional weights here and potentially also additional nodes here. So now you can think how many weights do you need to adapt for that? And I'll show in a minute that in fact it's actually not that many. So what we can therefore do, we just add those what we call adapters. We do a surgery in the network, both in the encoder as well as the decoder. And once we have this, we can then potentially also have additional query adapters. And the nice thing is, because we just add them to the input layer, as I'll show in a minute that this is usually sufficient, you can actually combine them, and they also nicely commute, so you get those nice properties. And then you can actually also share those adapters. So this way, you have a simple tool to do transfer learning, but only needing to adapt a few weights, which you can actually do at the user side. You can ensure privacy. You know, you never need to share those original samples, which with GDPR is, I think, a very important property, and which you also, for example, in this COVID-19 data set realize is important for us to have. Let me show you what you can do with this. This is a classic example for pancreas data integration. So we have a study here. We have here study labels shown by cell type, obviously nothing yet nicely integrated. If we now do this pre-training, you see cells integrate nicely across techniques and states, and you see sort of nice separation by the different states as you would have. Now you do a round of SCRs and add a new data set of a new measurement technique, in this case CellSig2, that hasn't been seen here before, and you see it nicely integrates again by study, except for this part here, and this in fact is, I think, alpha cells that the model here hasn't been seeing at all, and you see they nicely sort of attached to a different region. They don't overlap with the rest, as you would expect. Now you can do another round of adding and adapt, in this case for SmartSig2, and you see them integrate again nicely, and also those alpha cells you integrate with this newly added one. So you can build those references, you can extend building references as you would expect. Why do we choose those specific weights? Well, here's a bit larger data set, and in this case, 4 different brain RNA-seq expression data sets. And if we see how they cluster by cell type, by batch, we can see how homogeneous this is across batches and how homogeneous this is across in cell types. And if we only fine-tune those input layers, we get a good trade-off between those things that actually is not being improved if we fine-tune all weights. In fact, we can compare this by just adapting input layer versus all weights. We typically get a rather similar performance of this joint mixture of batch as well as cell types. And the number of parameters is a few order of magnitudes lower. That's why it's a good idea to just tune low dimension weights, not the full input layer and also not all weights. And we can also compare this to other batch integration methods that actually look at all the data at the time. You know, this SCR just does adapt this last layer, but if you even look at all data, you don't actually outperform these things. So that's why we argue it's useful and robust to have this very simplified regularization. I have a few applications, but I think I jump over the biggest one and just show you one application to these lung atlas data sets that I mentioned briefly before in the COVID-19 setting. In this case, we've been trying to map a lung lavage setting as a QA data set on a reference data set. And for reference, because we weren't sure if it should be only lung data, we also added PBMCs and bone marrow, just threw those things together, learned a joint reference. And then you see that the BARF integrates in some regions. We can actually see, in this case, it was BARF from patients with both a moderate as well as a severe COVID-19 state. And what you see is that it's not present all over, but it's present particularly here in macrophages. And if you zoom in what's happening in the macrophages, you do see that different factors are sort of acting in different places. In this case, this would be those T-cell resident macrophages that are actually strongly expressed in only the moderate COVID states, COVID-19 states, whereas those severe COVID-19 states were in particular active in CXCL10 positive parts, which are monocyte associated macrophages. So there's sort of 2 different types of mechanisms that you actually do see. And you see also a subtle thing that those moderate ones were mostly active also where the CXCL10 was active, so sort of an intermediate situation. And if you look at the immune cells, CD8, if you look at the CD8 positive T cells, you do see that there both disease states were mostly active in the lung situation, a bit different though, because there's an immune marker there. So in this case, you can nicely look at those data sets without sort of big effort to reanalyze and re-annotate those old things, and can just transfer annotations. And we have a few more examples on the webpage. Okay, so this is for model reuse. But can we maybe take one step further? And in this case, we've been thinking back to when we introduced ScanPy in the lab, that it really helped us homogenize the way we analyze, and we sort of put tools together, and we can sort of share things. But what we've realized with more and more of those data sets coming around, in particular, then method development needs to benchmark large scale. Can we help people improve benchmark, but then maybe also reuse and look at their results across scales in an efficient fashion? We aim to do essentially give easy access to annotated single cell data sets, and then also potentially trained models. This is what we call Spira. This is, I think, an idea of model zoos that has been around in genomics for some time, We've been trying to adapt this to the single cell world. It's work by two PhDs in that, David Fischer and Leander Doni, and we've just put it online now. We don't have bioarchive out yet, but please check out the GitHub and give us feedback if you have time. The idea is that we have a lot of these data sets readily available now. And what we did in Sphere, we just adopted a set of data loaders, many of those that download public data and give you annotations at the same time. And then you can embed them and you can visualize them. And we share those trained embeddings, so you don't need to retrain those. All of your UMaps essentially look the same if you apply this to this thing. But what you can also do, because we have a systematic framework for joint annotation that's consistent across those data sets, you can also do cell type prediction. You can annotate your own data sets quickly with that. And you can also add your own private data loader, so you don't need to somehow share your data on the top of one, but you can sort of do what we did with the SCRs. You can now do this across many of those public data sets. For this, we have an API. It's called the Sphere Dataset API. And for the different models, we have the model API that can essentially facilitate architecture use. So there you get quick access to data. Here you can get quick access to different types of tasks that you can come up with. And as a last point, those model parameters that you have here, you can just share again with cloud parameter storage with Zenodo, as we already pioneered with the SC Arches, and many other model tools have been doing before us. So with this, I think we have a set of easy accessible tools that give you both data set as well as annotation and model and parameters that you can share across. And with this, you can do some pre-learned embeddings. You have a versioning of weights and data sets. You can use them without any training, and you have a sort of very easy way of auto annotation. And this SC Arches model could be one of the models that you just put in here, but you can just use a plain old PCA and NMF or whatever you're interested in and apply this within this data set. It's quite large already. It has 240 data sets in there across 55 organs, both human and mouse, more than 3 million cells.
Left.
Thanks, I'm nearly done. You can, and that's sort of useful from a technical point of view. Usually you have those all separate, but you can now easily also stream a joint and data object. You don't even need to download it, but you can also stream. And as I said, you have for mouse similar dimensions. There's a way how this has been organized, but just as sort of for you to see how to get started, you just, for example, for mouse, you get some data set group for lung, and then you can just load the whole thing, or you have a streamed version of that, and you can then do your operation on top of that. Just to demonstrate how to quickly do this, we want to basically enable you to do quick benchmarks, try out your things, and also potentially include additional data loaders for that. We've been doing this with a few labs already, and it has been a lot of fun. But one application that we've been doing within the lab is a paper that Leander had out at the computational biology workshop of ICML just a few weeks ago. We've been trying to evaluate how important it is to put priors actually as additional precondition in those variation autoencoders that I showed you before. And here we compared different types of priors, in this case called event prior. I don't now say what it actually means, but just what Leander was quickly able to do is to compare this across many different situations because of those data loaders. In this case, he compared it across 16 public data sets, and he could give nice statistics. And I think if we are to move towards systematic evaluation of machine learning methods, I think we need those data resources. Computer vision has been, I guess, in parts transformed because it had this fantastic ImageNet available. And I think with HCA, LiveTime, and so on coming available, I think we need to have a quick way of sharing these things, and hopefully Sphere can contribute to that. With this, let me come to the conclusion. I've been showing you a bunch of tools from the lab. I encourage you to look at the GitHub if you have time. And just as an outlook, I think there's really fun things happening. Single-cell multi-omics is one of those directions that I now didn't speak much about, but I think we can imagine once you learn latent space across one thing, obviously you want to now try to include openness of chromatin, potentially some type of regulation and so on. And I think one of the nice ways that you can take this model-based approach of SCVelo is now trying to learn also interaction. Spatial genomics is one of the big next things coming up, where you can really get a spatially resolved picture of what your transcriptomes per cell essentially doing, and you can then learn really more robustly something about cell-cell interaction, cellular niches, a state, a tissue organization. And I think starting with the whole neural network and deep learning type of steps in those simple just a non-spatial model, I think enables us quickly to now scale up to also spatial models. I think neural network techniques will become more and more popular in that field. Last thing I wanted to say is with these interesting times of COVID-19, a lot of workshops become virtual. We've been in the single cell omics network, a German that I'm organizing, doing this similarly as well. And we've been organizing a workshop for temporal single cell analysis to really make sure to take into account time with a set of fantastic speakers from Dana, Alex, as well as Nikolaus, Julia, Caroline Uhler, machine learner from ETH. Really great set of people talking. If you're interested in that, it's free. Come and register and join us there. I think I acknowledged already all people from my lab. I thank you very much for your attention. This is sort of what things look like in pre-COVID times with our nice environment background. This is what Munich always looks like. If you want to come by. Thank you for your attention.
Thank you very much, Fabian. Fantastic talk. Quite impressive what you can get from a matrix of numbers if you take good care of the numbers. We have 4 questions in the lineup. I'll let a few seconds people to vote. We got 7 minutes, 8 minutes about, and you know that we get cut really at the second, so we will try to make this happen. First question, as a chair, we have a little bit of a privilege, and that's one of them. You mentioned at the very beginning, this cell type definition is somehow in debate. We used morphology before, we use RNA-seq recently. What if you start using velocities of the cell, for example, speed, et cetera, et cetera? Do we get an infinite number of cells types?
Yeah, I really like that. We have been going back and forth, and for me, I think, I mean, there have been even in early times, right? There has been, I think, this one paper from Dominic Grün and Alex, where they've been sort of zooming in again and again in one particular state that was sort of well defined, and then they were splitting up again. They saw them sort of even fractal-like coming up of new potential states. I think the question is similar to clustering. In a sense, it's a bit ill-posed because it always depends on the resolution of your problem, right? Let's say we have an organism. In our case, we look with Nikolaus Riesch, we look at this simple thing called flatworm planaria. That you can really get all the cells at the same time because it keeps producing those cells. You know, if you look at all of them, if you cluster them in a very coarse-grained fashion, you get organs. But if you're interested in, let's say, immune cells, you can sort of cluster them more fine-grained. If you may be interested in particular cell cycles, you get even more fine-grained. So in a sense, I think there's a lot of continuity that at some point needs to be somewhat randomly cut up for us small human minds to somehow be able to think about it. But yeah. I think it's not discrete.
Thank you. For the sake of time, let's go quickly. The most voted question is from David Bau. Do you think bulk genomics has become or is quickly becoming obsolete?
I do. I really need to be a bit controversial. I think even 5 years ago, so some of my colleagues in Cambridge, they've been using single-cell RNA-seq technique, just those protocols, as a cheaper version for doing bulk RNA-seq in low input samples. So, you know, they've been essentially just using the simple SMART-seq2 protocol, but they didn't just do it for single cell, but like for a few hundred cells. Thereby you average away effects such as cell cycling and so on, but then you get that. Having said that, of course, don't get me wrong. I think in many situations, let's say if you want to get clinically relevant data, sometimes it's important to get cell type resolution, then you do your single cell RNA-seq experiment. Maybe in many cases, it's not, because you can then do it computationally with deconvolution. So it's a good idea to throw a bunch of cells together. But I think the set of protocols with the miniaturization, with all the robotics and optimization that's coming, I think will really smear those fields out. And I think, yeah, we can't go back. So I think this massive multiplexing of experiments will be becoming a reality. We don't need those deeply sequenced bulk RNA-seqs anymore.
Good. Second question, more voted. Davides are popular in single cell. So it's Davide Cirillo this time. How much reproducible are the reconstructed cell lineages when the latent space is estimated using data sets of the same system, but from different experiments labs?
Yeah, great question. I think this is something that in early times we've been always struggling, and that's what I tried to really highlight here. If we don't use references, essentially each lab finds their own set of, not only, you ask about lineages, right? But let's make it simpler. Each lab even just finds their own subcell type or something like that. Each lab gets a different clustering. You know, in a sense, lineages are some type of continuous clustering or some latent space analysis. But I think the sort of more general thing is everyone finds a bit of a different latent space and then interprets them in a different fashion. We kind of need to agree on, as we did in genomics, what is our genes, what are our promoters, whatever. In this case, what are our smallest units to talk about? We need to have some type of ontology and map between those things. Human Cell Atlas would be one of those places where we can maybe find such an agreement. And there's an endeavor underway where we're actually setting up a common annotation platform. Peter Kachenko is guiding that, and John and myself and some others contributing to that, where we can actually have a multiple set of annotations present from different labs and then try to find a synonymous set of annotations. My simple approach at the moment is that we can just carry a set of annotations that we have here to another one using, for example, this architectural surgery or this aggregation of data sets that you can have in Spira.
Next question from Jose Maria Fernandez Gonzalez. The increased complexity of single cell experiments make me think that integrating the results from several of these experiments in a meta study is increasingly more and more complex. What do you think about this topic?
Yeah, thanks for saying that. I just had this super small thing in the end for our COVID-19 example where we had 160 subjects where we made, I think, what we called the first single cell meta study. But I think some other places are starting to do this. Monique van der Wiest and others trying to essentially do expression QTLs on a single cell level. So I think adding variation across population is one of the next big things, but it's tremendously complex. And you know, we need to be able to estimate mixed effect models with a million of samples in robust fashion. So these things, I guess, really need computational work. So I think it's fun for us, but I think this will happen and those data sets are starting to become available.
Thank you. Next question from Victoria Ruiz Serra. Is it possible to apply this to the protein scenario and perhaps complement the transcriptomics information?
Yeah, unfortunately, I sort of had to skip the last slide. I mentioned multi-omics being a big thing. And one of those first techniques that have been coming out in this regime is called site-seq and some variants of that, where essentially you take antibodies, tag them with an oligo so that you can make maybe tens or even hundreds at the same time, and then can measure RNA-seq as well as some protein expression. Similarly, of course, also techniques such as high-dimensional FACS or CyTOF give you protein variation. And I think tools for analyzing these are very related and similar. And I think getting this together also with proteomics becoming more and more low input, I think will be hugely interesting. And, you know, particularly this regulatory model thing that we try to do with this SCVLO dynamic model, adding now protein expression on top of that, I think is one of the next things. Leo Pachter's lab has already one of those papers out in this direction, getting some sort of protein velocity, but I think there's more to be done there.
We have 47 seconds. Diego Fuentes, how much of an influence does the initial training set of a transfer learning have on the overall result, and how different it might impact those results in the same query set?
It has a strong impact because this is going to be dominating the transfer. So this one typically reference should be large, but with those adapters, we show at least that the order of what you add in the end doesn't matter because we only adapt this first layer.
I think we'll leave it here. We had 2 questions left, but only 15 seconds to go, and I don't like when they cut us short. Thank you very much, Fabian, and thank you, the organizers of ECCB, for keeping this up even in COVID times.
Thanks for having me.
Bye, guys.
Bye.
Thank you.